Welcome back, everyone, to the deep dive. This is where we grab a whole stack of information, research papers, articles, maybe some notes scribbled down, and we just plunge right in. We pull out the key stuff, the insights that really matter. Basically, your shortcut to getting genuinely up to speed on something complex, hopefully with a few surprises along the way. So today's topic, it's something you've probably heard a lot about, maybe even got some in your kitchen cabinet right now. Omega-3 fatty acids. We're going to zero in on the long-chain ones, EPA and DHA.
Yeah, the ones you typically associate with fish oil.
Exactly. Fish oil, heart health, that's the popular image.
It is, and it's definitely a huge part of the conversation.
But when you start digging into the research, like the sources we've gathered for today, well, the science around omega-3s and, heart and metabolic health, it turns out it's way more complex.
You see these big studies, these clinical trials, and sometimes they seem to flat out contradict each other.
Right. And understanding why they get different results, that's actually where it gets, I think, really fascinating.
Absolutely.
So for this deep dive, we've got three main sources lined up. There's a fantastic, really comprehensive review article, pretty recent, that pulls together a lot of the current thinking on how omega-3s work and what the trials say.
It's quite thorough.
And then we're also looking at a couple of meta-analyses, which kind of pool data from multiple studies. One looks specifically at whether fish oil actually prevents coronary heart disease events, heart attacks and things.
Of course, a bunch of different trials, yeah.
And the other one, it digs into the safety side. What are the side effects people actually report in these omega-3 trials?
Which is obviously super important.
Totally. So our mission today is to navigate all this. We want to cut through the noise, the hype, maybe some of the confusion, really get into what these specific sources tell us about the latest on omega-3's cardiometabolic health.
Why those trial results can be so confusing.
Exactly. Explore the safety stuff and then try to figure out, okay, what does this detailed picture actually mean for you?
Sounds like a plan.
Okay, let's dive in and unpack this.
Right. So to get us all on the same page, let's start with the absolute basics. What are omega-3 fatty acids? Fundamentally, as the sources lay out, they're a type of fat, specifically polyunsaturated fatty acids, POFAs for short.
Polyunsaturated, meaning multiple double bonds in their chemical structure?
Exactly. And the omega-3 part, that just tells you where the first of those double bonds is located along the fatty acid chain. It's 3 carbons in from the end, the omega end.
Got it. Simple enough.
Now, there are a few different omega-3s, but when we're talking about heart health, metabolic effects, stuff we're focused on today, the attention is almost always on the long-chain ones.
Long chain, meaning they have more carbon atoms.
Precisely. The two main players, the ones you see mentioned constantly in these studies, are EPA, that's eicosapentaenoic acid, and DHA, docosahexaenoic acid. EPA typically has 20 carbons, DHA has 22. That length is actually important for how they fit into cell membranes and what they do.
Okay, EPA and DHA, the stars of the show.
Pretty much. And it helps to quickly compare them to the omega-6s. Another type of PUFA, you've probably heard of linoleic acid, LA, or arachidonic acid, AA.
Yeah.
Those are common omega-6s.
Right. Also fats we get from diet.
Yes. And like omega-3s, they're essential, meaning our bodies can't make enough on their own. We absolutely have to get them from food. The sources make it clear both omega-3s and omega-6s are necessary. But the balance between them and the specific types we eat, that can really influence health outcomes. That's a whole other deep dive, maybe.
Maybe. So focusing back on omega-3s, EPA, and DHA, how do we get them? Where do they come from in our diet?
Okay, this is a really key point the review article clarifies well. There are different pathways. First, there's ALA, alpha-linolenic acid. This is the main omega-3 found in plants.
Like flax seeds, chia seeds, walnuts.
Exactly. Those, plus things like soy, canola oil, some leafy greens too. Now, technically, our bodies can take that ALA and convert it into EPA, and then potentially convert some of that EPA into DHA.
So you can make EPA from plants.
You can, but, and this is a huge but, the process is really, really inefficient for most people. The sources stress this point. The conversion rate, often very low. We're talking less than 15%, maybe even less than 5% for DHA in some cases.
Wow, that's tiny.
It is. And it varies based on genetics, your sex, what else you're eating. Like if you eat a ton of omega-6s, that can interfere with the conversion enzymes.
Okay, so relying just on flax seeds and walnuts isn't going to significantly boost your EPA and DHA levels then?
Not practically, no. If you want to meaningfully increase the amount of EPA and DHA circulating in your body, the ones directly linked to these cardiometabolic effects we're discussing. The most efficient way, the sources say, is to get them preformed, consume them directly.
Right. Straight from the source, like seafood. That's where the fish oil connection comes in.
Precisely. Fatty fish are the classic example. Salmon, mackerel, herring, sardines, anchovies, trout. They eat algae that make EPA and DHA, and then it concentrates in their tissues.
So they do the conversion work for us, essentially.
Kind of, yeah. Or rather, the algae does. And then, of course, there are dietary supplements. Fish oil, krill oil, algal oil for vegetarians. These provide concentrated doses of that preformed EPA and DHA. The review really hammers this home. The distinction between ALA from plants and direct EPA-DA from marine or algal sources is critical. Most of the research showing cardiovascular benefits focuses on getting EPA and DHA directly.
That makes a lot of sense. You need the actual molecules that are thought to be doing the work. Okay, so we eat our salmon or take a supplement. The EPA and DHA get absorbed. What happens then? Where do they go, and how do they start influencing things at, like, a cellular level?
Right. This is where the biology gets really intricate, but also super cool. Once they're absorbed, EPA and DHA don't just sort of float around passively. They get actively incorporated into the structure of our cells.
Into the cell walls.
Into the cell membranes, yeah. They get woven into the phospholipid layer that forms the boundary of every single cell. Heart cells, brain cells, blood vessel cells, immune cells, all of them.
So they literally become part of us.
Exactly. And think about that cell membrane. It's not just a static bag. It's dynamic. Its makeup, especially the types of fatty acids in it, affects how fluid it is, how proteins embedded in it can move and function, how signals get transmitted across it.
Okay.
So by slotting EPA and DHA into those membranes, you can actually change the physical properties and the function of the cell. The sources even hint that EPA and DHA might do slightly different things here. The review mentions EPA might be better at preserving certain structural domains and preventing lipids within the membrane from oxidizing, like going rancid at a cellular level.
Unhealthy fats within the cell wall itself.
Yeah. Well, DHA seems particularly good at increasing overall membrane fluidity and influencing specific lipid clusters or domains. It's also highly concentrated in the brain and eyes, which points to its critical structural role there.
Interesting. So they're not totally interchangeable even at the membrane level.
Potentially not entirely, though the review notes it's still uncertain if these subtle differences significantly impact overall cardiovascular disease risk differently. But their structural role is fundamental, and that's just the start. They also influence cell signaling pathways. But maybe the most exciting area mechanistically, something the review really highlights, is their role as precursors.
Precursors, meaning they get turned into something else.
Exactly. They are the raw materials for making a class of incredibly potent signaling molecules called specialized pro-resolving mediators, SPMs.
Specialized pro-resolving mediators.
Yeah.
Okay, break that down. Pro-resolving.
Yes, that's the key word. Lots of people think omega-3s are just anti-inflammatory. And they do have anti-inflammatory effects, sure. But SPMs, molecules like resolvins, protectins, myrsins, derive mainly from EPA and DHA. They do more than just dampen inflammation. They actively promote the resolution phase.
The cleanup phase after inflammation starts.
Precisely. Think about it. Inflammation is usually a good thing initially. Your body responding to injury or infection. But it needs to be controlled, it needs to shut down properly, and the tissue needs to heal. If that resolution process fails, inflammation can smolder on and become chronic. And chronic inflammation is a major driver of diseases like atherosclerosis, buildup of plaque in arteries.
Okay, so SPMs are like the managers ensuring the inflammation process completes its job and doesn't linger destructively.
That's a great analogy. They actively orchestrate the shutdown. The sources give some cool examples from lab and animal studies. Molecules derived from DHA, like resolvin D2 and myrsin 1, were shown to block inflammatory signals, things like stopping the activation of NF-key, a key inflammation trigger. They also stop smooth muscle cells and artery walls from migrating and multiplying, which is part of plaque buildup. In animal models, these SPMs reduce the thickening of the artery lining after injury.
So stopping the bad stuff and promoting the healing.
Exactly. Other SPMs, like resolvin D1 and protectin D1, did similar things in other models: less thickening, fewer inflammatory cells showing up. Myrzin 2, another one from DHA, was shown to reduce obesity-induced inflammation. So these aren't just passive anti-inflammatories. They are active signals telling the body, okay, time to stop fighting, clean up the debris, send the immune cells home, and start repairing.
Wow. Okay, that's much more dynamic than the simple anti-inflammatory label suggests. They're not just putting a wet blanket on the fire. They're like directing the whole cleanup crew and the reconstruction effort.
Exactly. It's a very active pro-resolution role. Yeah. And this is considered a major way omega-3s, especially those leading to SPMs, might protect against chronic inflammatory conditions like heart disease.
That mechanistic insight is huge. It really paints a picture. Okay, let's bridge from these mechanisms to the effects we can actually measure, the impact on cardiometabolic health risk factors. What potential benefits do these sources point to?
All right, moving to the measurable risk factors. If you look across the sources, especially the review and trial data, the single strongest, most consistent finding, particularly when you get to higher doses, is the effect on triglycerides, TG levels in the blood.
Triglycerides, the fats we carry in our blood, often linked to diet.
That's them. And high triglycerides are a well-established risk factor for heart disease, part of what we call the lipid profile. The data is really clear here. High doses of long-chain omega-3s, typically in the range of 4 grams per day, significantly lower circulating triglyceride concentrations.
How significantly?
It can be quite dramatic, especially in people who start with very high levels, what's called severe hypertriglyceridemia, maybe TGs over 500 milli-GDL. In those folks, trials show median reductions of 30%. Even over 50% sometimes.
That's a big drop.
It is. And even in people with moderately high triglycerides, say in the 200 to 500 range, that 4-gram dose often brings levels down by 20–30% or more. It's a potent effect.
And do we know how they lower triglycerides? What's the mechanism there?
Yeah, the sources describe it quite well. It seems to be a two-pronged attack. First, omega-3s reduce the liver's production and secretion of VLDL particles, very low-density lipoproteins.
VLDL, those are the main carriers for triglycerides, right?
Yeah.
Shipping them out from the liver.
Exactly. Omega-3 seem to specifically target the larger, more triglyceride-rich VLDL1 subtype, telling the liver to make less of it. Second, on top of making less, they also seem to help clear triglycerides from the blood more efficiently, enhance plasma TG clearance.
So less going out and what's there gets removed faster. Makes sense why it's such a strong effect.
It does. It's probably their most well-established clinical benefit in terms of lipid modification.
What about other parts of the lipid profile, like LDL, the bad cholesterol, or HDL, the good cholesterol?
That's where it's less clear-cut. The meta-analysis that looked at safety and lab markers did report statistically significant decreases in non-HDL cholesterol and VLDL cholesterol, along with triglycerides. That fits, since those include the TG-rich particles. But for LDL cholesterol and total cholesterol, the results in that meta-analysis summary were pretty mixed. Some studies might show a small increase in LDL, some a small decrease. Many show no significant change overall.
So not a reliable LDL lowering effect like statins have.
Definitely not. The review mentioned some potential mechanisms, like maybe promoting cholesterol transport back to the liver or affecting ApoB production. ApoB is the protein marker on LDL and VLDL, but the overall clinical trial evidence summarized doesn't point to a consistent major impact on LDL or total cholesterol. The big lipid story for omega-3s is triglycerides.
Got it. TG lowering is the headline benefit there. What about inflammation beyond the SPMs we talked about? Any broader effects?
Yes, absolutely. Beyond generating those specific pro-resolving molecules, omega-3s have other anti-inflammatory actions. The review mentions they can improve the function of the endothelium, that's the delicate inner lining of our blood vessels.
Endothelial dysfunction. That's like an early warning sign for atherosclerosis, right?
It is. It happens when the endothelium isn't working properly, often due to things like obesity, insulin resistance, high blood pressure. It becomes less able to relax, more sticky for inflammatory cells. Cell studies showed DHA, for instance, could reduce the expression of adhesion molecules on endothelial cells. Those are the sticky Velcro-like molecules that grab passing inflammatory cells. Fewer adhesion molecules means a less inflamed, less sticky vessel wall.
Making the pipe smoother, less likely to get clogged.
Kind of, yeah. Omega-3s also seem to reduce the production of pro-inflammatory cytokines like IL-6 and IL-8, especially in settings of metabolic stress.
And how do they do that? Is it just by replacing other fats in the membrane?
That's part of it. By incorporating into membrane phospholipids, it displaces some of the arachidonic acid, which is an omega-6 PUFA, often used to make pro-inflammatory signaling molecules. Less raw material for inflammation, essentially.
Okay, so influencing inflammation through multiple routes, SPMs, endothelial function, membrane composition.
Right. And then there are effects related to blood clotting vascular protective effects. They can make platelets less likely to activate and aggregate abnormally. Platelets are essential for stopping bleeding, but when they get too sticky and clumped together inside an artery, that's what causes most heart attacks and strokes.
So omega-3s make platelets less sticky.
That seems to be the case. The mechanism involves EPA, in particular, reducing the production of thromboxane A2. Thromboxane A2 is a potent signal telling platelets to activate and clump. By reducing thromboxane A2 and potentially shifting the balance towards other molecules that inhibit clotting, like prostaglandin I, EPA helps maintain a less thrombotic state, less prone to dangerous clots.
Makes sense. Less sticky blood.
One source even speculated, based on some data, that DHA might actually be more antithrombotic than EPA, and that there could even be differences in how men's and women's platelets respond. Needs more research, but it's an interesting angle.
Okay. And any effects on blood sugar or diabetes? You mentioned metabolic health earlier.
Yeah, the sources touch on this too. There seems to be a link, possibly indirect, between the triglyceride-lowering effects and improvements in glucose metabolism. One meta-regression analysis mentioned in the review looked specifically at people with type 2 diabetes. It suggested that across multiple studies, omega-3 supplementation seemed to improve markers of glucose regulation and also reduce inflammatory markers like TNF-alpha and IL-6, which are involved in insulin resistance.
So potentially helping with insulin sensitivity.
Potentially. The review also highlighted a fascinating mouse study where giving EPA alone seemed to reduce insulin resistance, improve the function of the insulin-producing beta cells in the pancreas, lower liver fat, and even change the gut bacteria in potentially beneficial ways.
Wow, in mice at least.
In mice, yes. It's important to note, though, that the big meta-analysis looking at overall lab markers found mixed or non-significant results for standard diabetes, measures like fasting glucose and HbA1c across all the trials it included.
So maybe the effect isn't huge or consistent across the board?
It might be subtle, or maybe only apparent in specific groups or with specific formulations, or perhaps requires really rigorous trial designs to detect reliably for those particular markers. So promising hints, especially linked to TGs and inflammation, but maybe not a primary effect on fasting glucose for everyone.
Okay, so summarizing the potential benefits, really strong triglyceride lowering, some positive effects on other lipids like non-HDL, multiple anti-inflammatory actions, including active resolution via SPMs, improved blood vessel lining function, and making platelets less sticky. Mechanistically, it sounds fantastic for heart health.
It does build a very compelling case based on biology and risk factors.
Right. But this is where it gets confusing for so many people, myself included sometimes. You see all these potential benefits, this great mechanistic story, but then you look at the huge, expensive clinical trials designed to test the ultimate question. Do omega-3 supplements actually prevent heart attacks, strokes, and people dying from heart disease? And the results are, well, all over the place.
You've absolutely nailed the core issue, the central puzzle that dominates recent discussion, and is a major focus of the comprehensive review we looked at. How do we reconcile the strong mechanistic rationale and risk factor effects with the inconsistent results from the large cardiovascular outcome trials?
Yeah. Why the disconnect?
The review really dissects this by focusing on the contrast between two major highly influential trials, Reduce-It, Lent, and Strength.
Okay, tell us about those. Reduce it first.
Okay, Reduce-It used a high dose, 4 grams per day, of a very specific omega-3 formulation. It was highly purified EPA ethyl ester, called icosapent ethyl, or IPE, no DHA.
Just the EPA.
Just EPA. And they gave it to patients who were already taking statins but still had elevated triglycerides and were considered high risk. Either they already had cardiovascular disease or they had diabetes plus other risk factors.
So a high-risk group despite statins?
Exactly. And the result was pretty striking. Reduce-IT found a significant 25% relative risk reduction in major cardiovascular events compared to the placebo group. We're talking fewer heart attacks, strokes, procedures like bypass or stenting, cardiovascular death. It was a landmark positive trial for omega-3s, specifically for this EPA formulation.
Okay, 25% reduction. That sounds impressive. Now, what about strength?
Strength came along around the same time, also targeting high-risk patients on statins with elevated triglycerides and low HDL, a condition called atherogenic dyslipidemia. It also used a high dose, 4 grams per day.
Sounds very similar so far.
Very similar setup, but the omega-3 formulation was different. Strength used a combination of EPA and DHA, delivered as carboxylic acids.
EPA plus DHA this time?
Right, and the result, totally different. The trial was actually stopped early for futility. Yeah. The independent monitoring committee looked at the data and concluded there was basically no chance it was going to show a significant benefit over placebo for reducing cardiovascular events.
Wow. So same high dose, similar high-risk patients on statins, but one, EPA only, shows a big benefit, and the other, EPA plus DHA, shows nothing.
Exactly. That's the crux of the confusion. And the sources we reviewed spent a lot of time trying to figure out why these two seemingly similar trials had such divergent outcomes. Several key hypotheses emerged.
Okay, let's hear them. What could explain it?
First, the most obvious difference. The type of omega-3. Was it the EPA alone in reduced IT versus the EPA plus DHA combo in strength?
Could the DHA somehow cancel out the EPA effect?
That's one hypothesis the review explicitly mentions. It asks whether DHA might diminish or negate EPA's cardiovascular benefits. It stresses this needs more research, it's not proven, but it's a plausible idea given that EPA-only trials, like Reducide and an earlier Japanese trial called Gellius, tended to show benefits, while many EPA plus DHA trials have been neutral on major events.
Interesting. What else?
Second, maybe dose, though less likely here since both used 4 grams. But comparing to earlier trials with lower doses, maybe you need that high 4-gram dose to see outcome benefits, not just TG lowering. Yeah.
Third, and this is a huge point of contention discussed extensively in the sources, especially highlighted by that cohort study that tried to mimic the trials, the placebo choice.
The dummy pill they compared against, how could that make such a difference?
Well, Reduce IT used mineral oil as its placebo. Strength used corn oil.
Mineral oil. Isn't that pretty inert?
You'd think so, but the analysis presented in the sources suggests otherwise. The cohort study found evidence that the mineral oil group in Reduce IT actually saw their cardiovascular risk markers get worse over time compared to their baseline.
Worse? How?
Their LDL cholesterol, non-HDL cholesterol, ApoB levels, and even CRP, the inflammation marker, tended to increase in the mineral oil group.
Whoa. So the comparison group was actually getting slightly worse, which would make the EPA group look even better by comparison.
Exactly. It could potentially exaggerate the perceived benefit of the IPE treatment because the gap between the active arm and the control arm widened due not only to IPE's benefits, but also potentially due to the placebo's negative effects.
And the corn oil and strength. The data suggested the corn oil was much more neutral. It didn't seem to worsen lipid profiles, and it might have even slightly decreased CRP. So a more neutral or slightly beneficial comparator.
That's a massive difference in study design if the placebo isn't truly inert.
It's a significant controversy. The EVAPOR trial, which also used IPE and found it reduced plaque volume, also used mineral oil as a placebo. And in that trial, the placebo group showed plaque progression, while the IPE group showed regression, again suggesting the mineral oil wasn't neutral. So this placebo effect is a major factor considered in explaining the different outcomes.
Okay, that's a really important point. What are their differences?
Fourth, potential differences in the study populations, even though both were high risk. The review notes Reduce It had a higher percentage of patients who had already had a cardiovascular event, secondary prevention. Maybe the treatment works better or is easier to show working in that even higher risk group compared to strength, which had more primary prevention patients. Subtle, but could play a role.
Right, nuances and risk levels.
And fifth, something the review really emphasizes as critical, especially because older studies often ignored it, the actual blood levels of omega-3s achieved in the participants.
You mean how much actually got absorbed and was circulating?
Precisely. Just giving the same dose doesn't mean everyone reaches the same level. And here's a key finding. Despite both trials using 4 grams per day, the average circulating EPA level achieved in the strength trial participants, around 90 vml, was significantly lower than the levels achieved in Reduce It, around 144 mg/ml, and also lower than in the positive Gellius trial, around 169 mg/ml, even though Gellius used only 1.8 gg.
Wait, strength used the same high dose but got lower blood levels of EPA. Why?
Good question. Maybe differences in the formulation's bioavailability, absorption differences in the populations, baseline levels. Whatever the reason, the fact is the EPA levels in strength didn't get as high as in the positive trials. The review suggests there might be a therapeutic threshold for plasma EPA concentration needed to see cardiovascular event reduction, maybe somewhere around that 150 to 170 mg/ml mark achieved in Gellius and Reduce It. Strength didn't reach it on average.
And the dose needed to hit that target level might vary.
Exactly. It might take 1.8 grams for certain populations like in Gellius, but 4 grams of IPE for Western populations like in Reduce IT to get there. And crucially, many older neutral omega-3 trials never measured blood levels. So we have no idea if they failed because the drug didn't work or because they simply didn't achieve high enough concentrations.
Okay, that adds another huge layer of complexity. It's not just what you take, but how much actually gets into your system.
It seems absolutely critical for understanding these outcome trials.
So, summing up the reasons for conflicting results. It could be EPA alone versus EPA plus DHA. It could be the dose needed to be high enough. It could be huge differences caused by the choice of placebo. mineral oil versus corn oil, subtle differences in patient populations, and critically, whether the trial achieved high enough blood levels of EPA.
That's the multifaceted explanation emerging from these sources, yes. It really underscores why you can't just lump all fish oil trials together. The specifics matter immensely.
It really does. It moves way beyond a simple take fish oil message.
Definitely. And just to quickly mention that third source, the meta-analysis focused just on preventing coronary heart disease events. It sort of reinforces the mixed picture for general fish oil. Pooling data from various studies using different formulations and doses, it found no significant difference overall in death rate, sepsis, or angina between the fish oil groups and control groups.
So again, suggesting that while specific formulations at specific doses in specific people might work, a broad claim for all fish oil preventing heart events isn't consistently supported when you lump everything together.
Exactly. It adds another piece supporting the need for nuance.
This explanation of why the big trials differ is incredibly helpful. It makes sense of the confusing headlines. Okay, we've tackled benefits, mechanisms, and the outcome trial controversies. Let's shift gears to safety and side effects. What did that meta-analysis focusing specifically on adverse events find? What should people taking omega-3s, especially maybe those higher doses, be aware of?
Right. Safety is paramount. First off, the general consensus reflected in the review is that long-chain omega-3s are generally safe and pretty well tolerated. In the big trials, very few people, typically less than 5%, actually dropped out because of side effects. So for most users, they're manageable.
Okay, that's reassuring baseline.
But that doesn't mean there are no side effects. The meta-analysis that systematically combed through trial data for reported adverse events did find several things that were statistically more common in the omega-3 groups compared to controls.
What kind of things?
Gastrointestinal issues are probably top of the list, especially as doses go up. The meta-analysis found diarrhea was reported more often. The odds ratio was about 1.26, meaning roughly 26% higher odds of reporting diarrhea if you were taking omega-3s.
Okay, diarrhea. Anything else GI related?
Yes, constipation was also more frequent, surprisingly, with an odds ratio around 1.37. Nausea was borderline significant too. So yeah, general stomach upset, changes in bowel habits, nausea, these are relatively common.
The dreaded fish burps.
Well, related to that, another thing that came up strongly in the meta-analysis was taste disturbance, or dysgeusia. It had a really high odds ratio, almost 3.5.
Meaning people were over 3 times more likely to report taste issues.
Exactly. That could be the fishy aftertaste or just things tasting off. That can be quite bothersome for some people.
I can imagine. What about effects on lab tests? Anything show up there?
The meta-analysis did find borderline statistically significant increases in reports of abnormal liver function tests, specifically ALT and AST elevations, had an odds ratio around 1.35. Also some increases in BUN and hematocrit.
Should people worry about liver damage?
Usually, these are mild elevations noted in trials, not necessarily indicating serious clinical problems for most people, but it's something doctors might monitor, especially with high doses or pre-existing liver issues. The analysis also found statistically significant decreases in things like alkaline phosphatase, ALP, ApoB, platelet count, and TPA. Again, these are statistical associations from pooling lab data, not necessarily major clinical events confirmed elsewhere.
Okay. Now, the big one people often ask about with fish oil, bleeding. We know they make platelets less sticky. Does that translate to a higher risk of bleeding?
Yeah, that's a crucial question. Here's what the meta-analysis found. It did report a statistically significant increase in patients reporting something described as bleeding tendency. The overall odds ratio was 1.26, a bit higher for prescription formulas, 1.37.
So people felt they were bleeding more easily, maybe bruising.
That's likely what it reflects, yes. Maybe noticing more bruises, nosebleeds, that sort of thing. However, and the authors of the meta-analysis stress this limitation, based on the data they could pool from the included trials, they could not definitively say whether omega-3s increase the risk of actual clinically significant bleeding events, like major bleeds requiring hospitalization or transfusion.
Why not?
There just wasn't enough consistent, high-quality data on those specific severe events or on standardized measures like bleeding time across enough of the trials they analyzed.
Okay, so the feeling or reporting of minor bleeding bruising might be higher, but this specific analysis couldn't confirm if the risk of serious bleeds goes up. That's a really important distinction.
It's a critical nuance, yes. The antiplatelet effect is real, but quantifying the clinical bleeding risk from this pooled data was difficult.
Got it. Any other major safety signals that came out?
Yes. One significant finding, highlighted strongly in the comprehensive review and particularly noticeable in those large high-dose trials like reducing in strength, is an increased risk of atrial fibrillation, or AFib.
AFib, the irregular heartbeat condition.
That's right, an arrhythmia in the upper chambers of the heart that can increase stroke risk. Both Reduce It and Strength reported a statistically significant higher incidence of new-onset AFib in the groups receiving the high-dose omega-3s compared to their placebo groups.
That sounds serious.
It is a significant finding and a potential downside that needs to be weighed carefully against any benefits, especially for patients who might already be predisposed to AFib.
Do we know why omega-3s might increase AFib risk?
The mechanism isn't fully worked out, but the review mentions one hypothesis involving ion channels called piezo-1 in the heart's atrial cells. These channels sense mechanical stress and influence electrical activity. Omega-3s might interact with these channels in a way that contributes to the electrical remodeling that can trigger or sustain AFib. It's complex, but the clinical signal from the trials is quite clear and consistent across different high-dose formulations.
Okay, so increased AFib risk is a key safety concern to emerge from the big trials.
Definitely. And one last point from safety meta-analysis. They did note some potential differences between prescription versus generic over-the-counter omega-3s in terms of lab effects. Prescription ones seemed slightly worse for fasting glucose, while generic ones seemed better for HDL. Hard to know exactly what to make of that without more context, and they cautioned their overall analysis was mostly in middle-aged people with existing conditions, so safety might differ in, say, the elderly or during pregnancy.
Okay, that covers a lot on the safety side. So generally safe, but common GI stuff and taste changes. A signal for reported bleeding tendency, but clinical risk unconfirmed by this data. And importantly, a clear signal for increased AFib risk, especially with high doses.
That's a fair summary of the key safety points from these sources.
All right. We've been through the mechanisms, the benefits, the confusing trials, the reasons for the confusion, and the safety profile. Let's try to pull this all together now. Bringing everything we've unpacked from these sources, what are the main takeaways? What's the bottom line for someone trying to make sense of omega-3s for their health?
Okay, let's distill it down. First takeaway. Long-chain omega-3s, ETA and DHA, absolutely have plausible and well-documented biological effects. They get into our cells, they influence membranes, they have anti-inflammatory and pro-resolving actions, those SPMs. They affect blood vessel function, they make platelets less sticky. The scientific rationale for potential benefits is strong.
All right. The biology makes sense.
And second, for modifying risk factors, the evidence is clearest for triglycerides. High doses, like 4 grams a day, are undeniably effective at lowering high TG levels, especially very high levels. That's a solid clinical application supported by the data.
Okay, strong on triglycerides.
Third, when it comes to the ultimate goal preventing actual heart attacks, strokes, cardiovascular death, the evidence from large outcome trials is inconsistent. It's a mixed bag.
Some positive, some neutral.
Exactly. Trials using high-dose purified EPA alone, Reduce and Jealous, showed significant benefits in specific high-risk groups already on statins. But other large trials, particularly Strength using an EPA plus DHA combo, did not show a benefit. And some meta-analyses pooling various studies also find no overall significant effect on certain events.
So the preventing heart attacks claim is not universally true for all omega-3s.
It appears highly dependent on specifics, which leads to the fourth takeaway. Fourth, these inconsistent outcomes are likely explained by critical differences between the trials. The sources point to several key factors: the type of omega-3, EPA only versus EPA plus DHA, the dose used, the specific patient population studied, the controversial choice of placebo, especially the mineral oil issue, and crucially, the actual blood levels of EPA achieved. Hitting a certain target EPA concentration seems important.
All those details matter hugely.
Immensely. You can't just compare trials without considering all those variables. Fifth, safety. While generally well tolerated, common side effects are GI issues, diarrhea, nausea, constipation, and taste changes. More significantly, there's a clear, statistically significant increased risk of atrial fibrillation associated with the high doses used in recent large trials. The bleeding risk is nuanced, more reports of bleeding tendency, but unclear impact on major clinical bleeds based on the meta-analysis data.
AFib risk is a definite watch out.
It has to be part of the risk-benefit discussion, absolutely. And finally, sixth, what do current guidelines say? The review mentions that major organizations like the American Heart Association and European Society of Cardiology do incorporate omega-3s in specific situations. Based largely on trials like JELIS and REDUCE-IT, they recommend prescription-strength omega-3s, either purified EPA or certain EPA plus DHA combos, on top of statins as an option to lower triglycerides and reduce residual cardiovascular risk in certain high-risk patients who still have elevated TGs, while acknowledging and balancing that AFib risk.
So a specific recommendation for specific people under specific circumstances using specific products.
Precisely. It's targeted therapy, not a broad public health recommendation for everyone to take high-dose supplements for event prevention based on this evidence.
Okay. So the picture that really emerges from digging into these sources is complex. Omega-3s are biologically active, definitely lower triglycerides effectively, but whether they prevent heart attacks and strokes depends heavily on getting the right kind, maybe EPA alone, at the right dose, in the right person, achieving the right blood levels, and maybe even being compared against the right placebo in a study. And you have to weigh that against real side effects like AFib. It's absolutely not a simple fish oil cures all story.
That sums up the nuanced reality presented in these sources perfectly. The initial simple idea gets much more complex the deeper you dive into the actual evidence.
Well, this has certainly been a deep dive, and I feel like we've really unpacked the layers of this topic thanks to the insights from these sources. Thank you for joining us as we navigated the science of omega-3s and cardiometabolic health. We've seen the powerful biological mechanisms, the clear impact on triglycerides, the murky waters of the big clinical trials for preventing events, and critically, why they differ, and the important safety considerations, especially that increased risk of AFib. So as we wrap up, here's something to mull over, something to take away from this deep dive. We know that dietary sources of omega-3s, like eating fatty fish, are pretty consistently recommended as part of a healthy diet pattern. But the evidence for taking supplements, especially for preventing major events, is much more complex, pointing towards specific formulations, high doses, specific patient groups, and that AFib risk calculation. So the provocative thought is this. How does this detailed, sometimes confusing, nuanced picture acknowledging the TG benefits, the uncertainty around event reduction, the specific factors that matter, and the real side effects, how does that influence your thinking about getting omega-3s? Does it change how you view eating fish versus grabbing an over-the-counter supplement versus considering a prescription option? And how crucial does it feel now to actually discuss this complexity with your doctor, considering your own individual health profile and risks before deciding what, if anything, is right for you? Lots to think about there. Keep learning, keep asking questions, and keep diving deep. We'll catch you next time. That was the deep dive.